back if you're online, who are doing all this marvelous stuff for us. And I think we're good to go. So welcome back if you're online. So magically, we now have two people on the stage who are going to tell us about a key further partnership for Genomics England, and that's the commercial one and with world-leading researchers. And I have with me Parker Moss, who is Chief Commercial Officer of Genomics England, and Daphne Koller. And actually, I'd like you to introduce Daphne, Parker, so I'm going to leave that to you because she really needs a very big introduction.
She really does. I'm going to take this chair so that I can look at you directly.
Yes.
Daphne, it's such a pleasure to have you on stage. And in case there is anyone in the audience who doesn't know the litany of achievements of Daphne, I'm going to just run through them very, very briefly. So Daphne graduated at a ridiculously young age and became a professor in the computer science department in Stanford, where you were a much loved professor for 18 years. So that's a major commitment to education. You also, on the side, it seems, founded Coursera, which is this massive online educational community. I've done one of your courses, so you've enriched my life. Thank you for that. And that's now a very, very successful company. Daphne was the founding chief computing officer at Calico, which is Google's drug discovery company focusing on aging, and most recently has founded insitro, which is a fascinating drug discovery company that we're going to hear much more about. You picked up on your travels the MacArthur Genius Award, so you're a bona fide genius.
Oh, God.
And even more ingenious than that, you have become one of the latest research and also technology partners of Genomics England. We're so proud to have you here. Thank you very much.
Thank you so much, Parker, for that embarrassing introduction. And it's a real pleasure to be here, and it's an honor to be partnering with you.
Thank you, Daphne. So you started in computer science. I think you had a very broad kind of research aperture, but you focused recently on drug discovery. And what I would really love to know is why drug discovery and why now? Why did you decide to focus your research there?
So if I can go back a little bit, my interest in bringing together machine learning and biomedical data actually goes back over 20 years at this point, to the late 90s, when, as a machine learning researcher, I was looking for data sets that were both technically more interesting and also more aspirational than some of the ones that were available back then to machine learning researchers. And so I started working actually initially on some epidemiological data, seems very timely today. That was tuberculosis epidemiology, and then on the first microarray data sets, the first human genome, and so on and so forth. And always the challenge with doing the research that we really wanted to do was the lack of enough high quality data. I mean, at that point you felt privileged if you had a data set with a couple hundred samples. That was considered large. And we're now in a world where there is this abundance of data, which is only, I think, the beginning to what we're likely to be able to see in the coming years. And at the same time, of course, on the other side, we have this incredible set of machine learning methods that are able to make sense and extract insights from increasingly large amounts of data and many other areas of human endeavor. And so this seems to be a moment in time when those two tidal waves are about to come together in a way that I think offer us the opportunity to unlock some of the underlying secrets, the complexities that underlie human health and human disease. And maybe just to highlight where I think those data sets are coming from, I think they're coming from two quite distinct but yet synergistic sources. One, of course, is the human data, the clinical data, such as the one that you're creating at Genomics England and are also available in other places as well, with the growth, for example, of electronic health records and such. And the other is with the availability of increasingly sophisticated lab techniques that allow us to create in vitro model systems that are human-derived, human-relevant, and in a way that allows us to manipulate those systems, really start to interrogate disease and therapeutic interventions and try and predict, which is kind of what we aim to do at insitro, from the lab experiments and from the human clinical data, what will happen if we make a particular intervention in a particular human being.
Hence the name in situ, in vitro, and in silico.
Exactly.
Nicely concatenated. So you mentioned the value of big data. I'm the first to agree that big things are good. But I just want to drill into that a little bit more because, I mean, for example, at Genomics England, we're very proud of our colorectal cancer data set, about 4,000 patients, all with tumor normal pairs. Is a huge data set in life sciences terms. But it's small compared to the kind of things that you would have been playing with in academia, like ImageNet, that had tens of millions of data sets. So even in the large data sets that we curate, is that enough to power an unsupervised neural net?
So first, we believe the answer is yes, and that's for two reasons. One is that one of the techniques that the machine learning community has been able to really push forward in the last few years is the ability to use large amounts of data that are not directly relevant to the problem at hand and learn in some ways the language of images, and then undergo a process called fine-tuning, in which you take that representation, that language that you learned on cats and dogs and airplanes and things that have nothing to do with histopathology, for example, and use that as a way of creating that representation on which histopathology, a modest amount of histopathology, like 4,000 images, can be used to create a really robust representation. The other nice thing about histopathology is that, unlike images of cats and dogs, where each cat is an image, here the images are incredibly richly structured, and each piece of the image is in some ways itself a sample. You can think of each cell in some sense as being something that gives you more information. And so it's not as little information as it might appear just from the number 4,000 on top of what I said earlier.
So I think people often are surprised that cats and dogs come up in drug discovery conversations, but it is true through transfer learning that we really have learned to transfer some of the features of those images online, of people submitting their own pets online. That's one kind of technique that you've used. I know that something at the core of insitro is also your use of induced pluripotent stem cells,
yes.
IPSCs. And maybe you could explain a little bit about how that's an important part of generating this large number so that you can learn biological features.
So induced pluripotent stem cells, together with technologies like CRISPR and microscopy and single-cell RNA-seq, are some of the tools that incredibly talented life scientists and bioengineers have come up with in the last decade or so, and really been able to create a robust and usable toolkit that allows you to create in a dish effectively a diverse population of cells with different genetics, whether it's different genetics because these are iPSCs that are extracted from patients with different genetic burden of disease, or you could introduce driver mutations, for example, into those induced pluripotent stem cells and interrogate in a way that is a, N of one case control, which is you have what we call an isogenic pair, where you have the genome and the genome with the mutation, and you see what specifically this mutation does to an incredibly rich and multifaceted set of cellular phenotypes, so you can really start to interrogate in a human relevant system what each mutation does. So that genotype-phenotype function, which otherwise you're left to infer when you're dealing with natural variation, you're left to infer that from, do we have enough patients with the mutation, and are they sufficiently well matched to the patients without? Here you can really do that one-to-one experiment, and I think that really provides us with the ability to shed very deep light on what each of those mutations does, and maybe even illuminate some of those variants of unknown significance that we are currently struggling to identify and interpret.
So I think something that people struggle to conceptually understand is how you go from that kind of bottom-up look. In this, you mentioned iPSCs, but also I know that when you're looking at large images, you decompose them into small tiles or patches. How you go from that bottom-up view of a patient, but ultimately you're trying to make predictions about whole humans, their response to treatment, their prognostic predictions. So how do you collapse that very complex granular data into a prediction about the likely prognosis or response for a whole human in clinic?
Yes, so that is, I think, one of the really interesting things that we've been able to do is to move away from what is often done, which is rely on clinician annotations as a ground truth for training these machine learning models. Because what we found is that while clinicians have often a really wonderful insight about patients, they're also anchoring on things that they've been trained to look for. And oftentimes human biology surprises us with things that we didn't train our clinicians to look for. And sometimes those surprises are some of the most important insights as in terms of for instance, the identification of new therapeutic interventions. So we use an approach called weekly supervised learning, in which we don't train to the clinician annotations at the start. We take the images and we basically use a whole suite of machine learning tools that that we don't have time to get into, in order to learn without that anchoring the language of what we see in those images, the language of histopathology, the language of MRI data, the language of proteomics in the blood. And just by looking at the axes of variation in a natural population. And what we found, interestingly enough, is that if you have enough of those samples, even without that clinician annotation, in fact, better without that clinician annotation, you can now take a relatively small number of clinician annotations and learn oftentimes a very simple, what's called linear model that maps this language to, say, clinical outcomes of patients. And that when you do that, you sometimes find associations and insights that you actually don't if you were to train your models to the clinician outcomes at the start. And I find that to be a really exciting opportunity to kind of let biology speak its own language before you introduce that layer of human interpretation.
Interesting. So you're taking kind of a radically empirical view of biology thanks to large data sets, but it sounds like you are kind of elegantly interfacing human prior knowledge in as well in a linear manner.
Absolutely. And the other thing that we really like is we love using genetics as ground truth. So the ability to then take that embedding that we learned, or that language that we learned for histopathology, and ask, do we find in there associations to, say, genetic variants that oftentimes you don't see when you train to say the clinician annotations, and so the genetics in our eyes is in some ways a North Star to have we learned something that is truly meaningful.
Great. Well, we're going to go and talk about the kind of modality of genetics and ground truth in a minute. But before we go there, in the kind of challenge of drug discovery, you start off with target identification, and with all this data you have access to kind of exquisite target information. But we all know, anyone involved in drug discovery, that then validating those targets, saying something not just associative but causal about those targets, is such a challenge. How have you gone from just strong associations through your machine learning to making kind of causal inferences about drugs?
So this actually again comes back to genetics as sort of the core driver of our therapeutic strategy. You can get at causality via the genetics using techniques such as Mendelian randomization and others, because in human observational studies, genetics is the closest we can come to causality, because you don't get, by and large, to intervene in a human until the very end stage of drug discovery, which is clinical trials. So if you want to get at causality earlier in the process, which of course is absolutely essential because therapeutic interventions are intrinsically interrogating causality as opposed to just correlations. So that's one place where we get causality. The other place where we get that is, of course, via our creation of these in vitro model systems of human disease, where you get to introduce, for example, a candidate driver mutation or a candidate protective mutation into these iPSC-derived models and ask, does it revert the cellular system from what our machine learning tells us is a disease-like state to what our machine learning tells us is a healthy cellular state? And if that intervention does that, we know that that is something that is in fact a causal change, because we introduce that mutation into the in vitro system.
Okay.
And it's really, and of course the big challenge, and where we really like to push the technology is how do we gain conviction that the in vitro model system is truly predictive of human clinical outcome.
Right, and in a sense, that's how our conversation began, because at Genomics England, we focus on, in cancer, capturing the DNA and also the longitudinal clinical data of patients, but we've realized how important spatiality of the tumor microenvironment is alongside the molecular features of the tumor. And so we've started bringing imaging, whole slide images and histopathology and also radiology images into our data set. And we realized that we needed some help from a terrific engineering team like the one that you've brought together. So maybe you could just tell us a little bit about the partnership we've formed and what makes you excited about that.
Yeah, so we're incredibly excited about this partnership, and what we are basically building together with Genomics England is a multimodal search capability that allows researchers and clinicians to interrogate the Genomics England dataset in a way that leverages that language of, say, images that we've learned, and over time, as you accumulate other modalities, perhaps those as well, and being able to ask the question, here I have, for example, an image that maybe is unusual, or maybe I'm trying to understand what I'm seeing there. Can I find others that are like that? So if you look for pixel level similarity, you get complete nonsense. No two histopathology images are actually similar to each other at the pixel level, so you have to define a notion of similarity that in some ways is clinically meaningful. And that weakly supervised language that we've learned that really speaks to when are two things similar because they are relating to the same biology is something that allows researchers to create a cohort of patients that is similar in a biologically meaningful way and interrogate, for example, the genetics of that population. A clinician faced with a sample that they're trying to understand, I've never seen something like this before, what does it mean, can also look and see if there's others in the cohort that are similar to that, and maybe connect with the clinician who dealt with that patient and ask, what did you do with that patient? Do you understand what happened? Was the treatment beneficial to them, and so on? And so this is something that is beneficial, obviously, to researchers and to the patients that are part of the Genomics England community. And from our perspective, the data that's been accumulated in the Genomics England cohort is just an incredible treasure trove to hopefully identify therapeutic interventions that can be beneficial to patients down the line.
Yeah, it's a thrilling idea. And just to be clear to the research community that we're speaking to today, so we've just started work on this project together. This will allow researchers to find similar patients, but not based on phenotypic features, which is the usual thing, but actually based on sophisticated imaging features, which will be embedded in our data set. So we're working on that together, and then we're going to make that available to the research community as a resource. So we've just got a couple of minutes left, and I'd like to just slightly skew the conversation towards the participants that are listening today.
Yes.
You know, we've both experienced the clinical pathway ourselves, and I think one of the frustrations for participants is that drugs that are discovered today in cancer, for example, $2.5 billion to discover a drug takes 10 years, 95% failure rate. I mean, it's a crazy business model. Yes. How do you think that these new technologies and really your new philosophy in drug discovery is going to make lives better for patients?
So we believe that much of the drug discovery that has happened up until now has been driven by intuitions that scientists have had about, oh, this pathway, this gene might be implicated in disease, or sometimes even just serendipitous findings that happen by chance observation. We believe that the data that are currently being collected alongside the kind of much more systematic interrogation techniques for exploring those data can provide a much more rigorous approach for really testing hypotheses and asking, what do the data tell us about which therapeutic interventions are likely to be beneficial and for which patient populations? Because I think a lot of the failures that we're seeing is because many of the diseases that remain of unmet need are actually quite heterogeneous, and so effectively we're trying to cure multiple diseases with one therapeutic, and then we're surprised that we're not seeing the statistically significant signal that we're looking for. So hopefully the kind of really large, rich data sets that are being created alongside these machine learning methods will help point us in the right direction without being subject to the sort of hypothesis-driven and sort of oftentimes intuitive approaches that have led us astray.
So this is really a hypothesis-free approach to disease. I know you focus on liver disease and neurological disease. Maybe you can just tell us a little bit about your clinical focus in the future and also where we Genomics England fit in with that.
So the cohort that's been created here is wonderful in two ways. First of all, it creates for us an opportunity to hopefully make a similar type of impact in the oncology space, and so we're really excited about that. And then the rare disease cohort is, I think, one of the most exciting opportunities to truly interrogate that gene-function relationship, because oftentimes what you see in that cohort is the human analog of a knockout, effectively, a human knockout experiment. And if you could figure out for some of these patients, what is it that their disease is telling us about the function of individual genes, we can hopefully help not only they and others like them, but also learn something about that gene function relationship that might help people who have a less high penetrance version that is also driven by similar genetics, and maybe even help address a larger patient population.
It's a really exciting prospect.
Yes.
I can't believe we've already had 20 minutes on the stage. I could have spoken to you all day. Thank you so much for joining us, Daphne.
Thank you.
Thank you.
Well, that is a dazzling future laid out for us there. We're now going to have a break, and we're then going to go from our plenary to our themed tracks. So we're going to policy and initiatives, to cancer and rare disease. Rare disease will be back in this room. If you're watching online, thank you so much. You have a break to go and make a cup of coffee. See you later.